talk to Proshun, and I have changed the topic, and it's little different: microbial technology and vaccine design. So
Okay, sir.
let me share it. In microbial technology and vaccine design, actually nowadays the protection of the body is very important, and that's why we need the help from the microbial science. The people from the microbial science, they will give some idea, and they will help to design some vaccines, cause at a time epidemic, we have the experience about the corona. Epidemic can make a huge change, and for this reason the vaccine development is very important, not only for in India but also in world basis. It is very much urgent. Till date we have several vaccines to protect the disease, and out of them some diseases are eradicated, like the smallpox, and also some diseases are, I mean, in some country you cannot see those diseases, and in some of the country it's still there. Still it is rare, but we are looking for some vaccines, like the cancer, like the HIV, like the tuberculosis. So that's why the field is very important, and we need the help from the microbial science. In here, if you see, microbial technology is used in animal health and production in a number of ways, including testing, probiotics, microbiome modifications. Even this microbial technology, with the help of genetic engineering, can make your desired protein. And nowadays we are using a lot of the proteins which cannot be available from human being, but for the safety and the protection of human being, we are using these microbes. Out of them, identifying pathogens and parasite, microbial testing can help identify pathogenic and parasitic microbes in a farm and lab animals. Controlling disease, for example, mastitis, testing in cattle can help control the disease at the farm level. For the, I mean, animal benefit. Improving animal health, where a lot of farm animals are used for human being. They are probiotics, live microorganisms that can help animal digest feed, gain weight, resist infections. Antiviral and other drug preparations also require these microbial organisms. And reducing antibiotic use, so it has some side effect. So probiotics can help reduce the need for antibiotics in livestock. Improving animal health, as we have mentioned, if we know the cause of the disease and coming from the microorganisms, so microbiome modification can improve animal health. in domestic pets, farm animals, and wild settings. Improving food safety. Microbiome interaction can affect food safety. Also, follow-up is
Sir,
important. Yes.
sir, your screen is not given in the full screen mode.
Also, the slides are not changing, sir. Yeah, now it's okay. Slides are changing, but modes is not full screen.
Yeah, but the thing is that if I make it full, then it is not changing. I don't know why.
Okay, sir.
Okay, sir, no
Can
issue.
you see the change?
So now you are
Yes,
in
sir. We
slide
can
number
see the change.
3.
Okay, this is slide 1, this is slide 2, this is slide 3. Can you see the
Currently,
change?
you are in slide number 3.
You are on the slide number 3 only.
And now?
Still in slide number 3.
Okay, maybe there are some problems.
Now
So
it is changing.
Now it's come to 4. Now again 3.
again 3, yes. So if we go full screen, maybe some problems are there.
Okay, sir, no issue. You can continue.
Fools. I don't know how to change it. Is there any option to change it?
After making it in the full screen, we just go to the next slide. That is how we
Yeah,
operate
I'm trying to
generally.
do the same.
But there
Now
is
I'm in
some
full
problem.
screen, and then if I change, but you cannot see the changes. Okay. Anyway, here you have to increase your screen so that probably you can see full or in bigger zoom. Okay, reducing antibiotic use, like probiotics can help reduce the need of antibiotics in livestock and improving animal health, food safety, and improving carcass quality. Carcass quality is like when you are taking the flesh or from animals, different type of animals, either eat beef, chicken, pork, whatever, the quality improves if the infections you can protect in the farm, and then you can improve, like you can introduce some new protein as well as some amino acids which are available from this type of foods, specific amino acids. So in that way, carcass quality can be improved. Yeah, microbiome interactions can affect carcass quality. Biomining, microbes can oxidize metals, allowing them to dissolve in water, and chemotherapeutic purposes also, like antifungal, antibiotics production can increase. Now, microorganism as tools, you can see the transformation you have used regularly in your laboratory, that a particular gene can be introduced in a vector, and then that gene can be expressed in the cell, in the microorganisms, and then from there you can isolate the desired gene. Maybe you are not aware about the functions of that particular gene, but you know the sequence of that particular gene, introduce them, and finally can take out, and you can overexpress it and see the functions. So in animal model, you can introduce also, and then you can understand what is going on. Or this specific gene, you can introduce into vector, and finally, you can introduce into the germ cell to make an transgenic mice. So there in the egg, one pronucleus contain your desired gene, which you have make a clone and then introduce in the pronucleus, and then total egg you can retransplant into pseudopod mother animal, and then germ cell will express the specific gene, and after mating you can have a transgene or transgenic animal. So the same approach using, you can understand unknown function of a specific gene. Here the microorganism is the initiator, where you can make a clone of a gene and then introduce it into the animal. Now this part you know that I mean how transformation you can do, introduce the specific plasmid where your desired gene containing the plasmid with a promoter and a specific reporter genes are there, and after certain time, like for 40 seconds, jerking with temperature variation, the gene will be incorporated in the cells and then expressed in the media and selected by reporter gene. Now, another way production of the insulin, here you can see the human pancreas cell which actually produce this protein, human insulin. If you know the sequence of that particular gene from the bacterial vector, introduce that particular gene and recombine it, then introducing again bacteria, and it will produce this protein, this insulin. Now you can isolate this insulin, extract and purify human insulin. So it's going on. You are not taking the insulin from human being. Rather, you are taking only the sequence of that insulin gene and using this, microbes producing huge amount of this protein. And now components, here we are talking about the vaccine. There also you need the help of microbes. I mean, not all the cases you need the help of microbes, but most of the cases where this vaccine term refers to the preparation of live or inactivated organism or their antigenic constituents which have been formulated to stimulate specific immunity. So when you are thinking about the inactivated organisms, the chances of harmful effect will be less or zero. Why? Because the organism which you are trying to protect, like for example, these specific contagious organisms, and body should protect it once it will be exposed, and then immune system will be activated and then they will try to protect, but it will take some time, and depending on the incubation time, I mean the time between the infection and getting the symptom, in between this time is the incubation time, where incubation time is short, then the probability of harmful effect is high. Where this incubation time is long, then probability of development of disease is less. Why? Because within that period, maybe the body can activate its two branches of immune system, cell mediated and humoral. Obviously, the innate portion will work immediately. But when it is the organism is inactivated, is killed, they are inside the body, they cannot grow, they cannot replicate, they cannot duplicate itself. That's the benefit. And but their, I mean, whole epitope part is known, is open to the immune cells, and the immune system will be activated. Whereas when you are thinking about the live organism, then the advantage is you don't have to expose frequently to the immune system. Rather, one time exposure is enough because it is live and it will, replicate in the body. But the question is, if the live organism can enter in the body, it will try to replicate, and the harmful effect or the disease production will start. But when you attenuate the organisms, their pathogenic capability will be lost. Then probably they cannot develop the disease, or the chances of develop the disease is less or zero. When that happens, one time exposure is enough in the body, the organism is live and they will replicate. So continuously immune system can expose this epitope, and continuously effector cell as well as memory cell will be there in all the branches of immune system. And here antigens are very small amount of weak or dead pathogen that can cause disease. Very small amount means if the during disease development, the amount of pathogen can enter, that amount you can reduce and you can introduce. If it is live, you have to give the attenuated organisms, or inactivated organism you can use. They help the immune system learn this specific part like epitope and process it, activate the immune system, all the branches. Like the influenza virus is an example of an antigen, any type of bacteria, example of an antigen, and adjuvants here, which are in some vaccines are substances that help the immune system respond more strongly to a vaccine, because adjuvant can be added with the antigen, which antigens are very small in mass, but attached with the adjuvant. So they will be now stronger, more immunogenic, and activate the immune system. Now the vaccine benefits, two types of vaccines are there. One is therapeutic and another one is protective. Therapeutic means when the vaccine are used for therapeutic purpose, I mean already antigens are there and antigens are growing, developing in the body, and you want to protect the body from them, and there you use the vaccine. In another way, you or this type of antigen never exposed in your body, but you have given the part of that pathogen. So the immune system already known to, of that particular pathogen, and once in future the original pathogen can enter, they will protect it. So the protection purpose, this vaccine, most of the cases, vaccines are used for the protection purpose. And reduce exposure, I mean, the amount you can reduce, and also time to time, regular, you do not use these vaccines. And still, as the memory cell will be there, they will protect the body. And disease prevention by individual, by community, and producing the herd immunity. And also, very small price you can use to protect the total society, total community. And here, the herd immunity, I think I'm coming later how herd immunity works. Vaccine used for immunization, two types: active immunization and passive immunization. When indirectly vaccine can work or protect, it is passive immunization. When directly vaccine can protect the body, is active immunization. In passive immunization, method of acquisition include natural maternal, like baby or embryo is in womb, and mother is protecting the embryo because mother is already exposed different type of pathogens. And this pathogen, against this pathogen, mother's body already produces some immune product, including immunoglobulin G type. And only the immunoglobulin G, other than A, M, D, E, this G can pass through the placenta into the embryo. No other subtype can get entry through the placenta to the embryo. So these chances, I mean, the baby itself or the embryo itself cannot have the source of the immune system. It is not yet developed, but is protected. And this is passive. So antibodies, antitoxins, immunoglobulins from other animals can be there. Protection transfer from another person or animal, even like snake venom, one type of antigen exposed, and antibody against it from the horse can be introduced and will be protected. Now, for the active immunization, there, once the womb from the embryo is developed, is matured, then is delivered. Now all the hematological system already developed, and all the blood cells are now prepared, and in the peripheral blood, in the circulation, peripheral circulation, all the blood cells are available. And from the bone marrow, all the cells are producing. So there, the immune system will start to develop. But within that period, like until 3 months age of the baby, when the baby is delivered, after that, at least 3 months, it requires the protection from mother. And as it is detached from the placenta, from the mother's body, or through the placenta it was connected, but now there is no connection between the mother and the baby as it is delivered. So here the transfer of immunoglobulin from mother to the baby through the milk. And this milk is very important. That's why it contains so many nutrients as well as the immunoglobulin, which actually help the baby to protect from different type of pathogens. But after 3 months, or 1.5 months age, it starts to develop some immunoglobulin, and it requires the exposure of the antigen, and then the vaccine starts. Even the toxoid also uses there. And now they will produce the active immune cells and memory immune cells against specific antigen. And these are relatively permanent. So that in future, this time, as it is not exposed any type of pathogen earlier, so you can consider the immune system is totally blank or zero protection. But gradually in the environment, lot of pathogens are there. Some are too much aggressive pathogen. Some are very mild or poor to develop the disease. All types of pathogens are exposed in the baby. And now the immune systems of the baby start to develop the immune system. Now, heterologous vaccine is a term, is a type of live vaccines where one pathogen is introduced in order to provide protection against a different one. So, same type of pathogen has the epitope which can recognize the other pathogen too. Like different types of viruses contains the capsule, the character of those capsule or the epitopic structure of those capsule are almost same. In that case, heterologous vaccine is important. At a time, one vaccine can protect different type of viruses. The vaccines are pathogens of other animals that either do not cause disease or cause mild disease in the organism being treated, like Jenner's administrations of cowpox, vaccinia to protect against smallpox, BCG vaccine made of Mycobacterium bovis to protect against human tuberculosis. So different types of, though they one type of only the Mycobacterium bovis, but can protect different pathogen too. Now, warrant of passive immunization. When do you need the passive immunization? When deficiency in synthesis of antibody as a result of the congenital or acquired B cell defects alone or together with other immunodeficiency, then you need the passive immunization. When a susceptible person is exposed or likely to be exposed to a disease that will cause complication, like a child with leukemia exposed to varicella. Leukemia means its protective cells are defected. So their exposure of varicella or measles causes no protection and can induce the disease. So there you need the passive immunization. So this time they cannot permit for the protection, adequate protection. And when a disease is already present and the antibody may ameliorate or help suppress the effects of toxin like tetanus, diphtheria, or botulism. The toxin is antigen and it is already present in the body. So immediate you have to protect, otherwise it can the cell will be damaged where the toxins are there. So there passive immunization is important. Now, this passive immunization can occur naturally via transfer of material antibodies across placenta or to fetus for the baby. Injection with performed antibodies, human or animal antibodies can be used, like for toxin, snake venom. Injection of animal antibodies prevalent before vaccines. So sometimes it's like the animal antibody. Another one thing you have to remember that when you are giving the antibody from other animals, you can think about that this antibody also could be considered as antigen to the host. But that complementary site is very weak. There it cannot go longer. That's why it can be accepted by the host, though a poor reaction is going on inside the body. And effects are only temporary here. That's why passive immunization can be available from huge loss for the toxin or like venom, that type of things immediately. So this passive immunization can be helpful. Now, common agents used for the passive immunization, like for the black widow spider bite, horse antivenin; botulism, horse antitoxin; diphtheria, horse antitoxin; hepatitis A and B, pooled human gamma globulin; measles, pooled human immune gamma globulin; for rabies, pooled human immune gamma globulin; snake bite, horse antivenin; and tetanus, pooled human immune gamma globulin or horse antitoxin. These are used going on. Now, passive immunization with antibody transfer has some disadvantages. Though we use for the benefit, instant benefit, the transfer of antibodies will not trigger the immune system, though I have mentioned some complementary site of the antibody can cause idiotypic response, and a poor interaction can go considering this as antigen. There is no presence of memory cell, as it is not stronger, so that long-term effect will not be there, and no memory cell will be there when you are using this antibody from different animals. Risks are included recognitions of the immunoglobulin epitope by self-immunoglobulin paratope, what I have mentioned. Some individuals produce IgE molecule specific for the passive antibody, leading to mast cell degranulations. Here you can see the IgE causes allergy. So mast cell contains this IgE on their surface, and once the allergen cross-linked with this IgE, they will release different type of inflammatory factors like histamine, and then that can cause, it may be chronic or acute inflammations. So this is a side effect. Some individuals produced IgG or IgM molecules specific for the passive antibody, leading to hypersensitive reactions for the passive antibody. Now, active immunization. Active immunization is important because it can stay permanently for long term. Natural infections with microorganisms or artificial acquisitions, and that can contain these cells like the memory cells for long term, both stimulate the proliferation of T and B cells, resulting in the formation of effector and memory cells. The formation of memory cell is the basis for the relatively permanent effect of the vaccination. Two types of effect, as we have mentioned: immediate effect and memory effect. Immediate effect will eliminate the pathogen, and memory cell will store for long term and active for future infections with the same pathogen. So in that time, you are in relaxed mode, so that there will be no chance. If there is infections, it will be cleaned very shortly. Now, active immunization nowadays we have for the hepatitis B vaccine, diphtheria, pertussis, tetanus or DPT, and trivalent, savin or oral polio vaccines. So, and inactivated or shock polio vaccines. So, savin are live and shock are inactivated or dead. Now, measles, mumps, rubella, MMR, and Haemophilus influenza or HIV vaccines are available. Varicella zoster vaccines for chickenpox, and rotavirus vaccines for infant diarrhea, active immunizations. And effectiveness of vaccinations, like small percentage of recipient cell will respond poorly, role of genetic determinants, because due to the genetic determinants, all individuals have not the same genetic determinants. infections. And that's why vaccine will not be responded properly from all the individuals. Some of them can respond very poorly, but most of them, around 94% of the population, can work very well. So the immune system will be activated. And here the term herd immunity is there. So if 94% are activated and only 6% people in the populations, they don't want the vaccines, or they are not reacting against these vaccines, so the chances of transfer or transmit this pathogen or infections to those population is very less, and so the infection will not be there in this population, because most of them are have the activated immune system, and very small population they do not have. Still the chances is rare. When the small population they do not have, their infection is rare. Herd immunity, if the majority of population is immune, so chance of susceptible individual contacting infected individual is low. Now, herd immunity, factors affecting herd immunity, environmental factors are there, crowded condition, seasonal variation, strength of individuals' immune systems also depend on this, and infectiousness of disease, like greater the risk of infection, the higher the percentage of people need vaccines to attain herd immunity. And as long as the most of the people have this activation through the vaccines, the herd immunity will be produced. When enough people are vaccinated, chance of germ affecting the non-immunized population is small, can lead to disappearance of diseases like smallpox. Vaccination no longer necessary for the smallpox. Now, designing vaccines for active immunization. First factor, which branch of the immune system will be activated for this vaccine. So designers must recognize the important differences between the activations of humoral and the cell-mediated branches. Because once the humoral branches will be activated, immediate effect will be there, lot of antibodies will be in the serum, but these antibody will be degraded after certain time. Here, if you have the cell-mediated part, you have the memory cells. And especially for the viral infections, you need the activations of the cell-mediated immune system. And second factor is the development of immunologic memory. Failure to induce the formation of memory cell, leaving the host unprotected after the primary response to the vaccine subsides. So this is also important, that which part and whether memory cell is developed or not. If the memory cells are developed, so most of the, purpose of the vaccine development is done. Now, role of memory cell depends on incubation period of pathogen. Short incubation period like influenza, symptoms already underway by the time memory cells are activated. Because as we have mentioned, incubation period will be the period when the pathogen enters into the body and the time when it shows the symptom of the disease. So in between these, they can replicate inside the body. This is incubation period. So short incubation period is danger for the body. And repeated immunization with neutralizing antibodies for the short incubation period is required. Otherwise, you cannot eliminate the pathogen. And for the long incubation period, body will get some time to activate the immune system and can protect the body, like poliovirus. Enough time to allow memory B cells to respond, and there the pathogen, very chance to kill the pathogen by the direct immune system, whether it is memory cell or it will produce the memory cell and then will produce the humoral antibody, humoral response, like the production of antibody. Now, immunologic memory versus serum antibody, you can see when you are introducing the vaccine, immediately the serum antibody will reach within 15 days maximum. But after that, the antibody level will go down. And within 1 year, the antibody will significantly decrease. Whereas the memory cell will start to increase within 1 month, within 3 months, it will be in peak conditions. And it will go on for long term so that this memory cell can recognize the pathogen for future exposure in the body. And sometimes this curve can go down, and if then it requires the booster dose. So if it goes down, then again you can give the second exposure. So the memory cell now can go up again, and the second time exposure can goes huge up of the memory cell number. And in general, you have idea that one leukocyte can, the lifespan of a leukocyte when it is not exposed to any type of antigen in naive conditions, like 12 to 15 days. But when it is exposed any type of pathogen, it can be activated and their lifespan increases. And memory cell can stay for long term. Their lifespan will go more than a year. So in that way, the vaccine has the benefit to protect the body. Types of vaccine: whole organism vaccine, purified macromolecules, DNA, synthetic peptide, and multivalent subunit. Whole organism vaccine, either it is attenuated viral or bacterial cells. Attenuated means when you can grow them in conditions where it is not comfortable for them. Like the bacteria, the normal medium is not there to grow them. Rather, part of the normal media is there, and 12 to 15 generation, you can grow in different conditions. So their pathogenicity is lost, or very minimum pathogenicity is there. So in that condition, you can use the whole organism as vaccine. And inactivated conditions, in pathogenic, the wild type pathogenic bacteria or virus can be used by inactivated them with hot water, warm water, or by different type of chemicals. Now, purified macromolecules. So part of the pathogen can be isolated, like polysaccharide of bacteria can be isolated and can be used as the vaccine. Toxoid, so the toxin can be deactivated and can be used. So, or against it, the body will produce the antibody. So deactivated condition can be used as toxoid. Recombinant antigen. So part of the antigen, the pathogenic epitope can be introduced in a vector. So they can produce the protein which has the similar character or the epitope like the pathogen. And in the recombinant vector, there you can use the promoter of the vector and your required gene, which can be used as the epitope of the pathogen. Directly, you can use the DNA as vaccine. So I'm coming how DNA can work. Synthetic peptide and multivalent subunit also can be used as a vaccine in different way. Now, these are classifications of common vaccines for human, like bacterial cell, viral particle. In the bacterial cells like anthrax, cholera, pertussis, plague, and tuberculosis, typhoid. Tuberculosis and typhoid, like a live attenuated BCG can be used for tuberculosis, but it's not 100% successful to eradicate the tuberculosis. Still, in different country, you can see the disease. Typhoid, live attenuated by bacteria can be used. In all other anthrax, cholera, pertussis, and plague can be used as inactivated conditions. And for the viral particles, hepatitis A and influenza can be used as inactivated. And live attenuated conditions, measles, mumps, and polio, SABIN1, and inactivated SOC1 and rabies. Also, live attenuated can be used rubella, varicella zoster, and yellow fever. And purified macromolecules used for toxoid, for diphtheria toxin, tetanus toxin, inactivated exotoxin. And capsular polysaccharide for Haemophilus influenza type B or Hib, and Neisseria meningitis, and Streptococcus pneumoniae. And for surface antigen, hepatitis B is used for the vaccine. Now, designing of vaccine for active immunization, depending on the branch of immune system you want to activate, designers should recognize the important difference between the humoral and cell-mediated branches and development of immunologic memory. Now, whole organism vaccine, as we have mentioned, many common vaccines used consist of inactivated or attenuated cells, and included attenuated and inactivated vaccines will produce, but one-time shot requires the attenuated one. That will give the benefit not to have this booster dose. But here, one thing you have to remember that in general, when you are giving the attenuated one, that will be live, and you have to maintain the environmental temperature. Sometimes you need the fridge to keep it alive, otherwise it will not helpful. So if that facility is not available, so this attenuated will not work. But there is a chance it can revert back to wild type anytime, but that is very rare. Maybe in 10 lakh, one individual can show the symptom of the disease. Now, attenuated or bacterial vaccines, attenuation to reduce force, value, amount, or degree weaken, achieved by growth under abnormal culture conditions. BCG act as a double-edged sword, as they have distinct advantages and disadvantages. Disadvantages like is very rare, though they can revert back. And advantages is like one-time shot, continuously they will expose. And advantages like from their capacity of transient growth, prolonged immune system exposure, single immunization is enough, replication within host cells, that's why exposure will be continuous. Exception, Sabin polio vaccine consists of 3 attenuated strains of poliovirus. Colonization of intestine results in immunity to all 3 strains. Production of secretory IgA and induction of IgM and IgG results in the need for boosters. Individual strains interfere with one another. That will make a interaction inside the body, and hypersensitive reaction may occur. First immunization, and then one strain predominates in growth, so the other strain cannot show any exposure to the immune cells. Second immunization, immunity generated by the previous immunization limits growth of previously predominant strain, and that is the disadvantage. And third immunization, same principle as second immunization. So as first immunization, one will dominate and other two will not. And then the second immunization, maybe the second will dominate, other will not. In that way, third immunization can occur, can work. Now, disadvantages of attenuated. Major disadvantage is possible reversion, and rate of reversion of Sabin polio vaccine is 1 case in 4 million doses. Presence of other viruses as contaminants, because when you are producing the live vaccines, then culturing of this pathogen causes the contamination of other pathogen. And if it is not pure, then the other pathogen may start to develop inside the body. And unforeseen post-vaccine complication, some of the individual can show these complications. Now, inactivated viral or bacterial vaccine, inactivation by heat or chemical, by like formaldehyde or alkylating agent, and purified macromolecules as vaccines, like virulence of some pathogenic bacteria depends on antiphagocytic properties of their hydrophilic polysaccharide capsule. Coating of capsule with antibodies and complement increases the ability of macrophages and neutrophils to phagocytose such pathogen. And this can happen if that is, I mean, the antibody can attach with a specific protein. So that's why you can coat it, this part of the pathogen, like for the epitope purpose to the immune cells. And that will help, I mean, the phagocytic cells can engulf them and finally can display to the immune system helper T cells. Now, toxoid vaccines, and these macromolecules includes recombinant antigen vaccines, recombinant vector vaccine, DNA vaccine, synthetic peptide vaccines, and multivalent subunit vaccine. Here you can see the recombinant antigen vaccine or recombinant vector vaccines. Like recombinant vector, one, the vaccinia virus, their promoter is, you can, in the vector, you can take this promoter of the vaccinia, and then this your desired DNA encoding antigen can be introduced with this under this promoter, and then introduced there. They will be transfected in a tissue culture cells, and homologous recombination will make that particular protein, and you can select with the BUDR selection process, and the other will be dead, and only the portion you have introduced, that plasmid, which is, I mean, the incorporated the cells will survive, and they will produce that particular protein. So this is the vaccine as recombinant antigen vaccines or recombinant vector vaccines. And for the DNA vaccines or synthetic peptide vaccines, directly you can introduce the DNA, your desired gene, into the cells, like muscular injection. So muscle cell can incorporate these vaccines and this DNA, and then they can produce the protein, or peptide can be directly injected into the muscles. So after entry into the cytoplasm, through the endoplasmic reticulum, they will process and display on the surface with class 1 MHC. Then T cell will be activated, and through class 2 MHC, T helper cell will be activated, or the B cell will be activated. And once B cell will be activated, immediately they can produce the plasma cell or the memory cell. And the macrophages in the local portion where you have injected, they can also process these things, and they can interact with the helper T cells. The helper T cell will be activated, I mean, getting these informations from the macrophages, the antigen as well class 1, class 2 MHC. The helper T cell will activate it, and they can release some cytokines which can activate the B cells. So B cell can produce the antibody against that particular antigen. And in the cell-mediated branches, once the through class 1 MHC, the T cell will be activated, they can activate the cytotoxic T cells and memory T cells. So through the DNA vaccines, memory, both B and T cell memory will be produced. And now for the multivalent subunit vaccines, you can introduce different type of antigen in the solid matrix, and against it body can produce the antibody. So also different type of immune stimulating complex molecule with some lipid introduce them. Gradually they can be released, not at a time. They can be released gradually because of this lipid molecule outside. So once they will be exposed, in the cell surface, the TAP transporter molecule can take them in the proteasome, and this part of this peptide will be introduced in the endoplasmic reticulum, and they will synthesize this class 1 or class 2 MHC, and can be display on the surface. So in that way, the immune cells will be activated. So this is the multivalent, and here the advantage is that different type of antigen you can introduce here, which can show the epitope of that particular pathogen. So the T cell and B cell, both type of memory cell can be produced using these multivalent subunit vaccines. Now, the conclusion here you can see, in future we need new vaccines for cancer, malaria, AIDS, etc. Still we are waiting for that. New technologies for a combination and vaccines for non-infectious diseases may design new vaccines. A hybrid organism is created that produce the desired antigen, which is then used to make a vaccine. Multiple vaccines are combined into a single dose. Examples include combinations of influenza and COVID, and a combinations of influenza and RSV. Vaccines that are simpler to administer, survive transport even without refrigeration, and provide a more substantial and long-lasting immune response are needed. And that will be helpful for the remote area people who doesn't have the electricity, refrigeration. So that can help them to that population. The society will be benefited if that type of vaccines are developed. Okay, thank you. If you have any questions, I'm ready to answer.
Thank you, sir. You have explained a very important topic and in a very lucid manner. So I think our participants have gained a lot of knowledge from your presentation. Now we are open for questions. So if any of you have any questions, you can please ask. Sir, we have a request that can you please send your PPT to us? Some of our scholars, they are requesting for the PPT.
Okay, okay, I will send it.
Okay, sir. Okay, sir. Thank you. I think there are no other questions. So thank you for your valuable time once again for giving such a wonderful lecture. Thank you,
Okay,
sir.
thank you, Payal. Thank you, Prusun. I'll see you later.
So the next session will be from 12 p.m. It will be given by Dr. Obhik Shaha, who is the assistant professor in the Department of Health Science, Presidency University. So see you then. We will join at 12 p.m. again. Thank you.